connecting. All right, and I'm going to go ahead and share my screen. So please let me know if you can see it in a moment here. Still have some people coming in. Hey. Can everyone hear me okay?
Yeah, I can hear you, and I can see your screen too.
Okay, I'm going to put it on presentation mode here. All right, so let's go ahead and dive in. You've already heard a little bit of an introduction. My background is, as you heard, is in behavioral neuroscience. Attended the University of California, Berkeley, and I have worked in lots of different settings, as you heard. I'm currently head of a small biopharmaceutical consulting company. We focus on two areas of support for biopharma. Those include medical writing, and we'll talk a little bit more about what that entails. That is regulatory writing, clinical and non-clinical. And we also support a publication in scientific journals. The other area of support we provide is in regulatory project management, and that includes communication with the United States Food and Drug Administration. That's the health authority in the US that regulates and approves the use of drugs, devices, and biologics. We'll talk quite a bit in this session about topics that are specific to the United States, but I'll try to ensure that we also include a discussion of rest of world topics. And then I also want to point out that I am editor-in-chief of a peer-reviewed journal that is it's oversight of medical writers who are based primarily in the United States. So let's go ahead and jump into the agenda. These are the topics we'd like to cover over the next couple hours. One of those is the role of biopharmaceuticals in addressing neurologic and neurobehavioral conditions. So we'll just do a little bit of an overview of how biopharmaceuticals help to prevent and treat neurologic conditions. We'll also talk about how biopharmaceuticals are developed and how neuroscientists contribute to biopharmaceutical development. And there are three main areas where the contributions occur. Those are defined as discovery, preclinical research, and clinical research. So we'll get into quite a bit more detail about each of these topics as well. And then toward the end of the session, we'll tie it all together so you have a better understanding of the development process and the different activities of neuroscientists within that process. We'll also talk a little bit about opportunities for you in government, industry, and academia. So I really want to give you a sense of what some potential career opportunities are for you as you're completing your education and thinking about your future career. So let's start out with the role of biopharmaceuticals in addressing neurological and neurobehavioral conditions. And we're going to start with some basic definitions. So drugs, biologics devices, drug-device combination products, that's really what we're talking about. And just for a sense of efficiency, I'll generally just use the word drugs, even though I might be referring to biologics devices and drug device combinations. I want to make a main distinction between what a drug is versus a biologic. Most drugs are chemically synthesized, while biologics are derived from living organisms. So that's the distinction between a drug and a biologic. Most drugs have a specific known structure. Often biologics are complex and may have a somewhat variable structure. The important distinction for neuroscience is that biologics are generally very large molecules, and they generally do not cross the blood-brain barrier. And thus, biologics are rarely used in treating central nervous system conditions. So something to keep in mind is that mostly prevention and treatment of neurologic conditions is addressed through drugs and not through biologics. So here's just another way to represent the difference in biologic versus a drug. On the left side of the image is a drug molecule, ibuprofen, which is for treatment of pain. And the image on the right is a biologic. It's a monoclonal antibody. You can see it's substantially larger. And thus it isn't surprising that something that large would not cross the blood-brain barrier. In addition to drugs and devices, drugs and biologics, there are devices which can be used to treat neurologic conditions. We won't go into all that are on this list, but the bottom line is that a lot of neuroscientists who work in treatment of neurologic conditions work in the development of devices. So if you have a particular interest in engineering and in artificial intelligence that's being used, especially for development of devices, this might be an area that you would want to consider in terms of your future career. And then there are drug and device combination products. And these combine a drug or biologic with a device. And sometimes in neurologic conditions, they help to deliver a drug to the targeted area of the nervous system. So for example, if you have a large molecule, there may be a way to combine it with a device to get that drug to the central nervous system. There are various types of disorders that are being addressed through the development of drugs, and I'll just list out some of these. One area is neurodegenerative disorders. You're probably familiar with Alzheimer's, maybe Huntington's and Parkinson's. I've highlighted Parkinson's because we're going to use this as sort of the example condition throughout the presentation in terms of talking about treatment options. Another area being addressed by various drugs is neurotraumatic disorders, such as stroke and brain injury and spinal cord injury. Neuropsychiatric disorders is another category of treatment areas with use of drugs, device, and biologics, such as major depression, bipolar disorder, and we'll go through a few more types of disorders that are being evaluated, and we're attempting to develop better therapies for them. Neuromuscular disorders such as muscular dystrophy and ALS, seizures and infections. And I should mention too that there are various types of infectious agents such as HIV that get into the central nervous system, and so some treatments are directed toward those infectious disorders as well. There are some autoimmune diseases such as multiple sclerosis and inflammatory as well. So these are some of the key disorders that are being addressed by various types of drugs that are being developed. So we're going to spend a few minutes focusing on Parkinson's disease. This is an area that the foundation wanted me to describe in a little bit more detail than some of the other conditions. So Parkinson's disease is the second most common neurodegenerative disorder, And what we mean by a neurodegenerative disorder is that it has a progressive course. Typically, Parkinson's starts midlife to later. And there are primary symptoms such as tremors in the hands, arms, etc., muscle stiffness, slowed movement, impaired balance. And the symptoms usually start gradually and worsen over time. And that's typical for a neurodegenerative disorder is that they do tend to worsen over time. Some are more episodic, but Parkinson's tends to be sort of more progressive in a continuous manner. So one thing I'd like you to keep in mind as you study neuroscience is, and you may have learned this already in some of your coursework, is that the majority of the brain's activity is inhibitory, meaning that most of our synapses have an inhibitory effect on the downstream neurons. So why is that important? The reason that's important is much of what's happening in the brain is controlling and refining behavior, including muscle behavior. So in a disease like Parkinson's, where there's a loss of activity in neurons, that means there's a loss of inhibition, there's a loss of control of normally modulated activity. And that's, for example, why you see tremors in Parkinson's patients, is they have lost synaptic activity in a part of the brain that controls and produces smooth movement of your muscles. So always important to keep in mind that as you lose cells in the nervous system, often there's an emergence of behaviors that normally would be refined and controlled by the nervous system. So that's really what I've sort of described here. The nerve cells in a region of the brain called the basal ganglia. It's deep within the brain. There's death of those cells, and there's a reduction in the production of a neurotransmitter known as dopamine. There are some associated effects besides those that I described before. There are non-movement features of Parkinson's that include fatigue, irregular blood pressure, and decreased GI motility. So these are important to keep in mind, is that often these disorders have some primary effects, like in Parkinson's, primarily motor effects, but there are often associated features as well. There are some other aspects of Parkinson's that are important to keep in mind too. For example, in brain cells, we find that there's an unusual quantity of a protein called alpha-synuclein. And some patients who have Parkinson's disease, it appears to be hereditary. And in some of those cases, we can trace it to specific genetic mutations or variations. And many researchers now believe that Parkinson's primarily is a combination of genetic and environmental factors, including exposure to toxins. So there's still a lot that's unknown about Parkinson's, and there's no cure for it. But there are ways to detect it either through genetic testing or diagnosis through medical history and neurological examination. So we'll talk a little bit about treatment options for Parkinson's, but there are surgical options in addition to drugs and drug device combinations. So the drugs that primarily are used to treat Parkinson's, they increase the levels of dopamine in the brain. So really it's more of a, it's not restoring, of course, lost brain cells, but is meant to be supplemental to the dopamine levels that would normally be produced in the brain. And there's also an effect on other brain chemicals to help stimulate activity between nerve cells. The treatments for Parkinson's that help with motor symptoms also can help with the non-movement symptoms that we mentioned briefly. So there's a primary therapy for Parkinson's. It's called levodopa. It's a precursor to dopamine, which means that in the body, when levodopa is administered, it converts to dopamine. And the concern with a treatment like levodopa is that if there's a sudden discontinuation of the drug, it can produce rebound effects in terms of a patient's symptoms, such as impairment of movement and difficulty breathing. So this is a particularly challenging disorder, is that if you need to discontinue treatment, for example, because it's producing symptoms, side effects that are unmanageable, it can be very difficult to then wean the patient off of the drug because there are additional side effects they might experience just by going off the drug. All right, so that's a little bit of an introduction to some of the basic principles of drug development and how it relates to neurosciences. And now we're going to dive a little deeper into how biopharmaceuticals are developed and how neuroscientists contribute. And I should mention that the word biopharmaceuticals it's a mouthful. And a much easier way of saying that is just drugs, devices, biologics. You can pick what you want to focus on, but biopharmaceuticals is meant to encompass all of those different types of therapies that are used. Okay, we have a hand raised. Let's see. I think I'll need to wait because I'm not quite sure how to address that. All right, so let's talk about the different stages of drug development. The first stage is known as discovery. And that's where you're identifying potential drugs to target. So you're looking at your screening compounds to see if they have particular characteristics that might be useful for treatment of a disorder. And really, it's a screening process. So you're really looking at different candidate molecules to see, for example, if they might attach to a specific type of receptor in the brain. The process of screening for drug candidates it's a lengthy process, and typically there are thousands of different compounds that are screened. For every about 10,000 compounds that are screened, around 250 of those pass through screening and then are going to move on to the next stage of assessment, which is preclinical testing. So there's a lot of compounds that are going to be screened out. They're not going to be further tested. Small number proceed then to preclinical testing. So compound screening, there are different ways of testing these molecules. I won't go through the details, but robotics are being used a lot more now, and artificial intelligence is going to be an increasingly utilized method for identifying appropriate drug candidates. And so these are just some different ways that drug candidates are screened to identify those that might have the characteristics that should be evaluated preclinically and then later on potentially clinically. So let's talk a little bit about preclinical research. You've screened your compounds. Now you have some that look like they might work in your particular disease of interest. Preclinical is a term that is used interchangeably with non-clinical. So if you become familiar with preclinical research, you'll often hear the word non-clinical. They mean the same thing. So the purpose of preclinical research is to assess whether a compound has the potential to cause serious harm. So that's really the primary purpose of preclinical research, is to look at safety. Does it look like this compound is going to be safe? So before any compounds are tested in humans, they have to go through a whole battery or series of preclinical tests. And those tests typically are done both in vitro and in vivo, so in test tubes and in animals. And we'll talk a little bit more about some of the areas of preclinical research. There are three main areas of preclinical testing. One of those is called pharmacology. Another is drug metabolism and pharmacokinetics, which you'll see abbreviated as DMPK. And the final area is toxicology. And we're going to get into more detail on these three. So an important point here is that we talked about the 10,000 or so compounds that are typically screened to arrive at about 250 that make it to preclinical testing. Of those 250 that get into preclinical testing and go through a whole battery of assessments in the test tube and in animals, typically only about 5 of those continue on to the next stage, which is clinical testing. So there's lots of drugs that are being screened out through this process that appear to not have the right characteristics to move on to testing in humans. So something important to keep in mind. So let's dive a little bit deeper into each of the three main areas of preclinical research. And I will point out that when I started out in graduate school, I worked in preclinical research. You'll see when we get to the next slide on drug metabolism and on pharmacokinetics, that was my area of interest. Pharmacology is where you're assessing essentially the activities of the drug. What effects is it having on cells, for example, receptors? So primary pharmacodynamics is where you're looking at the intended effect of the drug. Are you wanting it to, for example, in Parkinson's, stimulate the production of more dopamine? Are you trying to produce an inhibitory effect? Secondary pharmacodynamics, you have to assess, well, what are some of what are called off-target effects? What are the unintended effects of the drug? So when you are developing a drug, it's not just going to have the one effect you're looking for. It typically will affect other parts of the bodies, and it may have some toxicities, some unsafe characteristics that you need to be aware of. Safety pharmacology is where you're specifically looking at which systems, organ systems in the body, a particular drug might be having an effect on. And you need to be sure that you're not adversely affecting different organ systems. You also need to assess combinations of your drug with other drugs to see if there are interactions. So pharmacodynamic drug interaction studies also are important to conduct as part of your battery of pharmacology tests. So that's one area of preclinical research. A second is in drug metabolism and pharmacokinetics, and that's where you're looking at the changes in the drug itself within the body. And you're really looking at various stages of activity and transformation of that drug in the body. Initially, it has to be absorbed. So, for example, if it's an orally administered drug, you need to know how is it going from the gut into the bloodstream. The distribution is looking at the movement of the drug from the blood to other parts of the body. It then is metabolized, so it's changing. It's being transformed in different tissues in the body, and then the last stage is it's excreted. Typically, so you're metabolizing, you're eliminating it. Typically, most elimination is through urine and feces. And commonly, what you'll see are pharmacokinetic curves that show the quantities of the drug in the blood. And so you create what's called a concentration time curve, where you're showing over time how much of the drug is measurable in the blood system. All right, so that's the second area of preclinical research. And the third that we'll mention is toxicology. Toxicology is where you're looking to see if there are dangerous effects of the drug. So you will do what are referred to as single dose toxicity studies. And what that means is you're doing ascending doses and giving single doses of that drug to an animal, for example, a laboratory rat, and you will test higher and higher doses until you see adverse effects in those animals. So that's looking at single doses. Then you will do tests that involve repeated dosing. So you'll want to know, okay, if this is a drug that needs to be used, we expect to be used long-term in patients, we need to know what are some of the effects of it when it's used over a long period of time. And so you'll typically do repeat dose testing in animals for weeks to months. And you look to see if there's some sort of longer term effect from repeated dosing. There are also studies you need to do to look at genotoxicity, meaning does the drug produce some changes perhaps in the animal's genetic profile or carcinogenicity? Is the drug causing abnormal growth of cells? And then you also need to look at reproductive toxicology. Is the drug potentially going to cause problems with fertility or problems with the development of an embryo or fetus? You also want to look at postnatal, meaning after birth development. If the drug is allowing the development of the fetus and birth after the birth of the animal, is there normal development or some abnormal changes occurring that might be related to the drug? And the final area in reproductive toxicology is looking at juveniles. So there's a lot of development that's occurring in juveniles of any animal species. You want to make sure that that development is normal and not being adversely affected by your drug. So typically there are dozens of non-clinical studies that are conducted. And here's just a few more toxicology types of studies where you're looking, for example, if you're administering a drug that is applied to the skin, is it producing any sort of irritation, immunotoxicity? Is it producing some sort of changes in your immune system and an ability to fight off disease? Some medications might have a risk of producing dependence. You also need to look to ensure that there are no impurities in your drug that might have negative effects as well. So as I was mentioning, you have a whole battery of these non-clinical tests that occur, and they are helping you to determine, should this drug move into patients? Does it appear to be safe? And does it appear that it'll be effective in human patients? So once you've gone through that battery of non-clinical tests, you're ready to go into clinical research. There's a lot to take in on this slide, so we'll go through it pretty carefully. Let's start at the top here where we have clinical trial phases. So it's important to know that when you get into human testing, you do your clinical trials, your tests in humans, in 3 phases. And not surprisingly, they're referred to as phases 1, 2, and 3. So let's start with the phase 1 testing. So just in terms of, to give you a little context, a phase 1 study typically lasts 1 to 6 months. They typically involve healthy participants. So you want to test first in healthy participants just to get a sense of, does this drug appear to be safe? That's really the main assessment you're doing in phase 1. If there are side effects, what are those side effects? Maybe you get a bit of an idea of how the drug works in the body, but the primary purpose of phase 1 is simply to test that and show that the drug is safe. And you're often looking at different dose levels to make that determination. So when you've done your phase 1 testing, usually that's in a given drug program, typically there's 1 to 10 phase 1 studies. The number of drugs that move on from phase 1 is typically about 7 out of 10. So there's a pretty high success rate of drugs getting through phase 1 and then into phase 2. So let's talk a little bit about phase 2 testing. So these are longer studies. They typically are 3 to 18 months, and they involve patients with the condition of interest, typically 10 to 50 participants. This particular slide was based on a condition called fibrodysplasia. So this is specific to that condition, but depending upon the disease area of interest, you can have a small number of participants. Some conditions, such as cardiovascular conditions, you may have hundreds of patients in phase 2. So you're also looking to make sure the drug's safe. Doing some preliminary testing to assess whether it's effective. And then also you're assessing how much of the drug should be taken and how often. So you're looking at different dose levels and different dosing intervals. So should the drug be taken once a day, every day? Should it be taken 3 times a day? Should it be something that's only once a week? So you're doing all sorts of assessments, often based on pharmacokinetics of the drug, knowing how much of it's in the bloodstream over time to help you determine how often the drug needs to be taken. So once you've gone through your phase 2 testing, typically there are multiple studies there. About 1/3 of those drugs make it through phase 2 testing and then move on to phase 3. So phase 3 are your large studies, long-term often. Typically, the studies are a year or more, and often they have dozens to hundreds of participants, sometimes thousands. And you may have anywhere from 1 to half a dozen phase 3 studies, typically, depending upon the disease area. So in these large studies, you're also assessing whether the drug's safe. You're trying to determine how effective it is, what kinds of patients it's most effective, and are there benefits or risks to specific subpopulations? So for example, is the drug problematic for elderly patients or patients with kidney disease? So you're looking at specific subpopulations to see if there's any additional benefits or risks to those subgroups of patients. And from the compounds that go into phase 3 testing, about 1 out of every 4 make it through phase 3 testing. On the right-hand side now, you'll see that once you've gone through all that testing, you, as someone developing a drug, need to provide your data for regulatory review. So what does that mean? Regulatory review is assessment by the health authority in your country, and they will determine whether they consider the data from your trials to justify approving that drug to go into patients on the market. So the data are collected from your studies. You analyze the data, you describe it in documents, and you present those documents to your health authority. In the US, it's the Food and Drug Administration. In Europe, it's the European Medicines Agency. Various countries have different health authorities that will assess the data to determine whether they will allow that drug to be marketed in their country. So the health authority ultimately makes a decision. Yes, you can market this drug in your country, or no, we don't consider it safe. We do not approve it. So this is really the sort of final stage in terms of taking all that data you've gathered, both in your preclinical setting and in your clinical setting, presenting that to your health authority, and then they decide whether the drug is going to be approved. All right, so let's talk a little bit about the role that neuroscientists and others play, specifically in clinical research. One of those areas of involvement is what's referred to as program strategy. And I will tell you that there's any one of these topics I'm about to describe, a person could involve their entire career in supporting that one bulleted item. So, for example, some individuals work specifically in developing a program strategy for an entire drug. So they will determine, okay, here's the drug that we want to evaluate. We need to decide what kinds of studies we're going to do, what kinds of patients we're going to look at. Are we going to compare our drug to other drugs that are on the market or that are commonly used in patients? So program strategy is one area of involvement of neuroscientists and others who are thinking kind of big picture about how to assess a drug that is going to be developed. Drilling down to studies, each study that you conduct has to be designed. And we'll get into more details on what we mean by that. It's an area of clinical research that a lot of people are involved in designing individual studies. For those who have a particular interest in data, biostatistics and statistical programming are areas that are important in clinical research. And so we'll talk a little bit more about those in a bit. Data management. So typically there's large amounts of data we're collecting in a given study, and we have to determine not only what data we're going to collect, but how are we going to collect it and manage it in a database. So data management's an area of involvement of neuroscientists and others. There's also an area called project management. So this is the complexity of clinical research is substantial, and there are typically dozens to hundreds of people involved in a clinical research program, and you have to ensure that everyone's moving in the same direction, that everyone is aware of interdependencies between projects, timelines, who's involved. So project management is part of ensuring that these complex programs are moving along efficiently. And clinical operations is the actual planning and conduct and implementation of the studies. So if we're going to do a study in hospitals and we're determining that we need to test this drug in lots of different countries, clinical operations is responsible for ensuring that the program is conducted out consistently in each of the countries, that there are personnel who are involved in each hospital that are familiar with the study and know how to conduct it. So clinical operations is a big part of conducting clinical trials. Let's spend a few minutes talking about different types of studies in clinical research. So we had talked in preclinical about pharmacology, DMPK, and toxicology studies. In clinical research, there are also various types of studies. One type of study is referred to as a clinical trial. And this is the most common type of clinical research study. This is where you're testing new treatments for diseases. And that's done, as the name implies, in a clinic. There are also studies that are referred to as natural history studies. That's collecting health information to understand sort of the disease in a population and how it changes over time. There are what are referred to as biospecimen studies, where you're looking at tissue samples, could be also blood-based. And then there are surveys. It's another way of conducting research, asking questions of patients to determine whether, for example, a particular behavior might be related to a higher incidence of a disease. All right, so those are some types of trials. Let's talk specifically about trial designs. So there are various types of clinical trial designs, and sometimes those combine multiple elements. So I'm going to give you an example of a type of trial. What I'm referring to here is multi-center, randomized, double-blind, placebo-controlled crossover study. So it's a lot of elements. Let's break that down. So let's say we're going to conduct a study. We want to conduct it not just at one trial site. We want to conduct it at multiple centers. So that's referred to as a multi-center study. Randomized, it means we're assigning patients to one of two treatments or one of three treatments. So we're just randomly assigning them to the different treatment groups. Let's just say two treatment groups. Double blind means that neither the patient nor the research team knows what treatment that patient's been assigned to. Have they been assigned to active drug? Have they been assigned to placebo? So placebo-controlled is one type of way of assessing a drug to see, does it have a noticeable effect that differentiates it from patients who are on placebo. You might also do your double-blind study against an active comparator. That would be a drug that also works in these patients. You want to see, does my drug work as well as that active comparator, maybe better? So that's one way of assessing it, is in a double-blind active controlled study. And crossover study means that each patient is getting one of the treatments initially and then the other treatment after that. So they're crossing over from, for example, active drug to placebo, or they started out with placebo and then they go on active drug. And that's just one way of ensuring everyone gets active drug in a study. There are other important design elements that might be applicable, such as prevention studies. Do we want to see, if we administer this drug in patients, can we prevent the onset of that condition? Another type I'll just pull out here is meta-analyses, where you are, let's take a dozen different studies and let's pool all of that data from those studies, do a meta-analysis, and see if that gives us a better picture of the properties of that drug in terms of safety and efficacy. An important aspect of conducting clinical research is selecting appropriate patients for your studies. And in every study, you define what are referred to as your eligibility criteria. You want to include certain types of patients. You want to exclude certain types of patients. And usually in clinical studies, you're highly restrictive in the types of subjects you're allowing into your study. And why would you do that? Why would you be highly restrictive? You want to have a fairly homogeneous, tightly defined population, because you want to remove a lot of the variables that might make it difficult for you to determine whether your drug is safe and effective. So initially, the studies have a very narrowly defined group of patients, and thus results from clinical research may not be generalizable to a broad population of patients. So often a drug will go through a series of clinical research studies with a fairly narrowly defined set of patients, and it's not until the drug is actually on the market and in the general population that you start to see, does it work better in certain types of patients than others? Are there safety concerns in a particular subpopulation that we weren't aware of when we did clinical research studies? So it's important to identify appropriate patients so that you can test your study endpoints. And endpoints, it's an important concept in clinical research, because this is how you're measuring the effect that your drug is having. So an endpoint might be, for example, in Parkinson's disease, is my drug reducing the frequency and severity of tremors? So that would be an endpoint. One would be frequency of tremors. The other would be severity. You might also have safety endpoints, such as is this treatment for Parkinson's reducing gastrointestinal motility or activity, or is it actually improving activity of the gut? So it's important to define your endpoints. And often in these clinical studies, there are dozens of endpoints. You'll typically define one endpoint as your primary endpoint. Sometimes you'll have a co-primary endpoint where it's one efficacy endpoint and one safety endpoint. And then you typically will have several secondary endpoints. Those are considered slightly less important. And then you may have some exploratory endpoints where they're not really key to your research thesis, but you still want to assess some other types of endpoints that might be important in the future. Once you've done your study, you need to analyze your data. So you're going to collect a lot of data, and there are various sources. You may be collecting it through electronic health records, registries. You might be doing surveys. In clinical trials, typically data are collected on what are called case report forms, where the clinical staff are entering the data onto forms. Usually they're electronic or ECRFs, electronic case report forms. And then that data goes into a database. Sometimes the data are what are referred to as quantitative, so they're measurable and distinct. Sometimes the data are qualitative, meaning there's sort of a range of experiences that a patient may have. If a patient is having better quality of life, and they can sort of measure that and say, yeah, on a scale of 1 to 10, I would say my quality of life right now is a 7. That can be useful information. These are referred to as patient-reported outcomes, or PROs. And often studies now are using patient-reported outcomes as an important assessment of whether a drug should be approved and made available to patients. Because ultimately, you want to be improving patient life, patient quality of life. So it's more and more often now, these patient-reported outcomes are being used in clinical research. I mentioned before that the data typically are captured on case report forms. Typically, those now are electronic. It used to be you filled them out with a pen, but now you just enter the data directly into a form, and that goes into a database. So the data analysis it takes the involvement of biostatisticians and statistical programmers. So let's break that down a little bit. Biostatisticians are, they're really thinking about the design of the studies. What are the endpoints that are important to include? What are the appropriate analysis methods? Once the data are analyzed, they're produced in the outputs are referred to as tables, figures, and listings, or TFLs. So that's where you're actually going to see the numbers on paper in tables, figures, and listings. The biostatisticians will look at the data and help to interpret those numbers. So often there are dozens, sometimes hundreds of tables, figures, and listings that come out of a study, and you really need someone with an understanding of the data to help interpret what it means. That's typically the biostatistician helps substantially in that regard. You also need a statistical programmer, someone who actually writes the program to analyze the data to create the tables, figures, and listings. So these are a couple areas where biostatisticians who have a particular interest in data sometimes then end up specializing in biostatistics or statistical programming. So there are a number of different types of methodologies to analyze clinical trial data. And we'll just talk about a couple of them here. I'll focus here on descriptive statistics, and then we'll talk a little bit about survival analysis. So descriptive statistics is providing a sort of straightforward analysis of the data. Let's say we collected data in Parkinson's patients, and we were measuring the severity of their tremors. And the severities were measured on a scale from 1 to 10. And we were looking at their severity of tremors over time. Let's say it was a year-long study. So over time, you might have done your assessments every month, and you want to look month by month. What was the average tremor score? What was the mean? What was the median, which is half the values are above, half the values are below? How much variance is there in the data? Were most of the patients around a 5 with very little, variation from 4 to 6, or was there huge variation from 1 to 9? And it's really sort of hard to tell whether the drug is going to be effective in a large number of patients, because some it looks like it works and others it won't. So that's looking at descriptive statistics. Another type of analysis would be what's referred to as a survival analysis. This is done for disorders that often have a fatal outcome, such as in cancer studies. You want to evaluate the time to an event. So in cancer studies, for example, you want to know how long are we preventing the patient's recurrence of cancer, or how long are these patients typically on drug before they die. So you're doing what's referred to as a survival analysis to look at survival rates and disease progression. All right. So we've talked a bit about some areas in clinical research where neuroscientists and others are involved in really a breadth of different activities related to conducting clinical trials. There's another area of drug development that sometimes people in the neurosciences go into, which is referred to as chemistry manufacturing controls. So if you have a particular interest in chemistry, this might be an area of appeal to you. CMC is the shorthand for this area. And this is where you're focusing on how you're going to manufacture and control the drug that you're developing. So there are two aspects to the manufacturing of drugs that are important to distinguish between. One is the development of a drug substance. So that's the active substance that then is packaged into something that's delivered to the patient, which is called the drug product. So you have to ensure that you're not introducing impurities, that every time the drug substance and the drug product are being manufactured, that they have the same characteristics. So you want to ensure that there's control of that drug quality. So these are important aspects of developing and manufacturing and controlling the drugs that are eventually going to go into patients. Another associated area of clinical research is referred to as regulatory affairs. So people who work in regulatory affairs, they are the individuals who are involved in developing the documents that are going to go to a health authority to get that drug approved. They also make sure that the company is complying with national and international guidances regarding drug development. They're making sure that you're monitoring safety of the study drugs and also drugs once they get onto the market. And regulatory affairs teams also are involved in evaluating the safety and efficacy data that are coming from the trials. and inspecting the manufacturing facilities and how the drugs are being distributed. So regulatory affairs, one of their primary responsibilities, as I mentioned, is helping to obtain approval for the drug to be made available to patients in a particular country. Typically, the drug approval process, one country is targeted initially, and then once the drug is approved in that country, that package is package meaning the documents, including data, are then passed along to other countries to pursue approval in those additional countries. So it is an interesting process where, again, it's often approval is sought in one country initially, and then the drug moves on to other countries. There's often a very US-centric, I guess, focus on drug development because a lot of drugs initially are approved in the United States, and that typically makes it easier for the drug to then get approved in other countries. All right, so let's talk a little bit about some additional functional areas that are involved in clinical research. One of those is medical writing. I mentioned that's one of my areas of focus for my company. And we provide regulatory writing support, which means writing, for example, the reports that describe a clinical research study or that summarize a collection of studies. So that's referred to as regulatory writing. The other area I mentioned is publication writing. So if you have a study and you want to ensure that the scientific community at large is aware of that study, the results from that study, you would write up an article, submit it to a journal. They will review it, and if they accept it, then your research will be published in that journal. So we provide that kind of support as well. So that's referred to as scientific publications. There's ongoing safety surveillance and what's referred to as pharmacovigilance of drugs. That's looking at, as the data set gets larger and larger, meaning more and more patients are being evaluated and have received a drug, you want to continue to assess whether there are new, what are referred to as safety signals. Is there a trend that's emerging in patients that wasn't seen when you had a smaller number of patients who'd been treated with that drug? So there's ongoing assessment of safety, not only in your clinical trials, but also once the drug is out in the general public and is being marketed. Marketing is the area, and this marketing, of course, applies to many other areas of commerce, which is what are we going to do to ensure that this drug gets to the targeted population, and making sure that physicians and patients are aware that this drug has these properties and might be able to help the patients. And then there's an area called medical education. So this is, you're not promoting the drug per se, but you are educating caregivers and patients in regards to the properties of this drug. All right, so we've covered a lot, and I'd like to kind of tie it all together now in terms of talking about the development process from beginning to end. So we started off by talking about discovery, how we're looking and screening lots of different compounds to see if they might have the right characteristics that potentially would be useful in patients. Those, though, first will go into preclinical testing, where you're looking in the laboratory setting in vitro and in vivo at the characteristics of that drug, and then determining which ones should move on to clinical research. So you'll conduct your phase 1, phase 2, and phase 3 studies. And once those are completed, you will write up your data package, submit that. Here we're referring to FDA review, but really it's health authority review, whatever country we're talking about. That health authority then will determine whether they approve your drug. And if they do, you will continue to monitor the safety of that drug post-marketing, meaning once the drug is already out in patients. So let's kind of recap the whole development process from discovery to approval. We mentioned at the outset that there are, let's say you are screening 10,000 compounds. Over the course of screening of those compounds, typically about 250 of those would be identified for going into preclinical testing. The average time frame for going from initiation of screening through to the end of preclinical testing is about 6.5 years. So it's a long process. Once those compounds go into clinical testing, you will have typically 5 of those compounds that were preclinical will go into clinical trials, and the process of going through phase 1, 2, and 3 testing typically takes about 7 years. There's some range to that, but it's on average 7 years. Once you have completed your clinical trials and you've written up your summary of your data and you've submitted that to the FDA, they have or health authority in general, they have a review period, often, well, so typically from 6 months to a couple years. They will review the data. They often will have questions of the researchers that require responses, and then they will determine whether they're going to approve that. So at the end of this process, often you'll just have one drug approved. So we've gone from screening of 10,000 compounds, 250 that went into preclinical testing, 5 that went through clinical trials, and of those 5 that went before the FDA, typically only one of those will get approved. You can see the span of time for this process averages about 15 years. So it's a lengthy process. In addition, it's very costly. So people who are involved in drug development, think of it as a high risk, high reward industry, because there's a lot of failures and there's a very high cost to develop a drug. So let's focus on the right-hand side of this slide. So the cost of developing a drug, let's start back in the 1970s, where it costs an average, and this is US figures for development of a drug, it costs an average of $179 million to develop a drug in the 70s. You can see that the breakdown was most of the cost was related to preclinical testing. In the 1980s, costs were on average $413 million to develop a drug. Still, non-clinical research is the majority of the costs. And in the 90s and into the early 2000s, the cost to develop a drug had jumped up to a little over $1 billion. Now, with the clinical research costs being higher than those of the non-clinical. So health authorities generally requiring more human data than before, and that's accounting for more of the costs. From the 2000s to the early 2010s, the cost to develop a drug had increased to an average of $2.6 billion. So a huge, huge cost to develop one successful drug. That represents a 145% increase from 2003 to mid 2010. So it's a big investment. Once a drug is approved, there's also what's referred to as post-approval development. So there are additional studies that might be conducted to look at other patients, populations you might want to treat with your drug. You might look at different formulations. You might look at different dosage strengths. An average of $300 million is spent on post-approval development as well. So looking at the post-marketing a bit more, some of that cost is related to what's referred to as post-marketing safety surveillance. So you do need to continue to assess whether your drug is safe in the general population. You're not doing clinical studies anymore, or maybe you're doing some additional studies, but really your focus is on marketing your drug and looking at safety in the general population that's receiving the drug. You might be doing some expansion efforts to look at additional types of patients that might benefit from your drug. So that's marketing and expansion. So let's take a case product example. So there's a drug called Sinemet. This is a Parkinson's drug. It's a combination of carbidopa and levodopa. So this particular formulation, it provides a controlled release of carbidopa and levodopa. So often, once a drug is developed, you'll see that over time, researchers will look at different formulations of that, different strengths of it, maybe in combination with other products. In this case, also looking at a way to sustain the release of the drug so that you don't have to administer it. So there's more even delivery of the drug in the body than if you give it in a sort of pulse manner, where a patient has a dose of the drug, it has its effect and wears off. Then you give a dose of the drug, effect and wears off. Here, there's more of a sustained release. So these patients who receive Sinemet, it's particularly useful for helping with their slow movements, so helping to increase the speed of movement and to reduce muscle stiffness. Is also helpful in dealing with the tremors and swallowing problems that are characteristics of patients with Parkinson's. Now, this drug was originally approved way back in 1975. It's now available as a generic. So something to keep in mind is that in many countries, there's a limited patent life for a drug, meaning that once a drug is approved, the sponsoring company or researchers have a limited number of years to make money off of that drug to recoup their costs of developing it. And then after that patent expires, that means other companies can make generic versions of that drug and sell it to patients. So that is beneficial, of course, to patients in that once they're generic, the costs go down because now there are multiple companies making the drug, and that competition drives down the prices. Okay, so now we're going to shift to our last topic, which is opportunities in government, industry, and academia. And hopefully over the course of this discussion so far, you've gotten some feel for the range of areas that you might work in if you have a particular interest in taking your neuroscience career into a sort of biopharmaceutical development direction. So we'll spend a few minutes talking about opportunities in 3 different sectors: government, industry, and academia. So what are some of the opportunities in government? Depending upon the country, there are often research facilities that are funded by the government to conduct research or to provide funding support to independent researchers to conduct their research. So sometimes grants are provided to individual researchers or to consortiums or not-for-profits. So people who work in government may be, for example, assessing proposals for research grants and helping to decide who's going to receive the funds from the government to to conduct their research. Industry is another area where a lot of work in biopharmaceutical development is occurring. There are over 20,000 biopharma companies in the world across more than 200 countries. So there's a tremendous amount of research being conducted in industries. And there's also a lot of research being conducted in academia. Academia, the difference there typically is the focus is not on making sure that you are developing something that's going to be profitable. It's truly sort of advancing research, and often it's done in conjunction with industry or government to provide a funding source. So even though we talk about these three different sectors, government, industry, and academia, often they're very much intertwined and helpful to each other in terms of helping advance assessment of different drugs to treat various conditions. So now we're going to do a little Q&A session. And I will show you on this next slide some resources. What I will provide, I'm going to provide to the association this slide deck so that any of you who might be interested in following up on any of these resources, or if you want to contact me through either of the email addresses provided here, you are welcome to do that. So I'm going to stop sharing at this point, and let's see if I can ensure that we can now open it up to any questions that the group might have.
Thank you so much for presenting to us. That was like an awesome presentation. I took so many notes actually. So there are some questions. Someone asked that about like what your advice would be for someone wanting to study in the field of behavioral neuroscience and what like some careers would be in that specific field.
In behavioral neuroscience? Sure. So that's actually what my PhD was in, is behavioral, and my focus initially as a student was on animal behavior and hormones. So looking at aggression, so the relationship between hormones and aggression. So the question is in regards to sort of how to explore behavioral neuroscience as a career. So if you decide that you want to go into an advanced degree program, and often the best approach is to look at the different degree programs in various universities and see if there's a program that describes a curriculum that's most appealing to you. So for example, if you're interested more in behavioral neuroscience with animals or with humans, then you can be directed to a program in one of those areas. In my case, I found after doing work in animal research that I've stumbled upon clinical research. My advisor was contacted by a research clinic, and they were looking for a graduate student to conduct neuropsychological assessments on patients who were in a drug treatment trial. So this was a trial where they wanted to know, does this drug produce any adverse effects on a patient's speech, memory, and so I was conducting regular assessments of patients over time to see if their cognitive performance was holding steady or getting worse, or perhaps even improving over the course of treatment. And that gave me a great interest in clinical research, and so I shifted my focus from animal research into human applied clinical research. So it was just sort of a happenstance because I had never even heard of clinical research before being contacted by this clinic. So that was something I was hoping to do with this presentation, is to give all of you a picture of some of the different areas that you might go into that you would not normally be exposed to as a student. And I'm not sure I'm quite addressing the question, but this is really sort of intended to give you a feel for some avenues you might take that, again, you wouldn't necessarily learn about in school.
And then another question was like how AI helps in screening out drugs in preclinical testing.
Yeah, so I think the AI is just getting a preliminary start in helping with compound screening, which is helping to assess. So, for example, one of the key characteristics of screening compounds is, and let's just take a straightforward example, is this molecule going to fit in this receptor? And AI can be used to help determine whether that shaped molecule is going to fit in the receptor without having to actually test it in a laboratory setting. So, again, we're at the very beginning stages of AI assessment of how it can be used for compound screening, but this is one of the ways where you can take a library of thousands of compounds where you have a known chemical structure and then saying to AI, help determine whether this is going to fit this receptor. So that's one example.
And then like what types of animals would be used in drug testing?
Yeah, that's a good question. So in animal testing, we're using mammals. We want to ensure that the animals are selected, that they are going to, the effect in the drug on those animals is going to be similar to what we would expect in humans. And so we know, for example, that there are characteristics of certain rodents where we can use them as a pretty good early approximator of how the drug is going to work in humans. So typically, rats, mice, guinea pigs are used in research. And then in some cases, we're assessing in dogs and in primates, monkeys typically. There's more limited use in terms of monkeys, and usually those are limited to small toxicology studies. So the majority of studies are done in rodents, smaller number done in dogs, and then least commonly in primates.
Okay. And then also, um, there's just like a clarification question. So is safety pharmacology about assessing side effects?
Yes, so right, because it is about side effects. And I realize there's some overlap between that and toxicology. Um, but safety pharmacology um, is looking at quantities of the drug, where it's going in the body. But yes, it's often sort of has overlap with what you're assessing when you're doing toxicology studies.
And how do you differentiate between the reproductive toxicology types and like what are the key differences between them?
So in the different types of toxicology studies, all right. So um, one of the types of toxicology studies is looking at fertility. So if we give this drug to female rats, are they able to then become pregnant? Some cases you find out, oh, this drug reduces fertility. You also will want to look at in pregnant animals, if you administer the drug, is it causing the fetus to abort? Is it producing changes in the fetus? And then if there's a live birth, is it a healthy birth? Are there any changes in that are negative in that animal after it's been born? And you're looking also sometimes at juvenile animals to see are there longer, is there sort of a delay in negative developmental effects that might be occurring that we should be aware of in juvenile animals. So you're kind of looking all the way from ability to of an animal to get pregnant, all the way through to are there negative effects, all the way through to development as a juvenile.
And then also like a question was like, why would people be willingly a subject to like a drug trial?
Yeah, that's a good question. Okay, so let's start with healthy volunteer studies, or those are the first phase one studies you do are typically in healthy volunteers. Why would someone do that? Well, honestly, there's typically a financial incentive. Patients are healthy subjects, I should say, are compensated for participating in a clinical trial. An effort is made to ensure that they're not going to have any negative effects from the drug, but there are possibilities that they're going to have side effects. So the motivation, frankly, is typically financial, that someone wants to participate in a research study to make some money. Now, there are what are referred to as institutional review boards. These are oversight groups that make sure that the studies that are being proposed are not going to present an unreasonable risk to study participants. And so they also want to make sure that there's not an enticement or inducement that's excessive for people to participate in something that might be risky to them. So that's healthy volunteer studies. That's usually the motivation is for financial reasons. In patient studies, often it's patients who truly want to determine whether there's something that might work better for them. Or they recognize that participating is going to be helping to develop something that might help people in the future. So there's somewhat of an altruistic element to it as well. And sometimes healthy volunteers do it for that reason as well, that they simply want to help contribute to building some knowledge about whether a drug might work.
And a question I actually had was like, I feel like I've heard of a lot of like problems in drug development, like big pharma kind of thing. And so I was wondering like what main problems that you think are like the most important
Yep.
about drug development?
Yeah, that's a very fair question. And there's some people who are involved in I would say the vast majority of people I've come into contact with in drug development, whether it's in government, not-for-profits, academia, or in industry, they are doing it for the right reasons. They want to advance knowledge, they want to help patients, and so their motivations are very pure and positive. There are folks who are involved, particularly in the biopharmaceutical industry, where their motivation is profit, and their main interest is in making money. And that's where the industry gets a bad reputation, and sometimes deservedly so, is that some folks who are making decisions in companies are doing it solely with an interest in making money, and they're not being driven by the desire to help patients and to improve science. So that's kind of the what I've taken from a career in the field is that most people are in it for the right reasons.
Okay, thank you.
Hmm.
And then another a question that somebody had was how how companies get like financial backing while
Yeah,
conducting research.
yeah, that's a good question. I mean, that's one of the biggest challenges, of course, in developing therapies in general, is the high cost to do the research, as you saw, hundreds of millions to billions of dollars to conduct a research program. And how do you pay for that? So there are two main ways that companies generate the funds. One is through private investment, meaning venture capital firms, for example, will say, we will provide you with $200 million for your research, and you will pay us back when your drug is on the market, and then we will get X amount of the profits from it as well. So they're venture capital, meaning they are providing the funding for the research to be done with an intent that later they'll be paid back, and plus plus profit. The other mechanism is through companies that are public companies, meaning you can buy stock in those companies, and that the funds from the stocks are used to pay for the research. So you'll see that just as within other industries that are on the stock market, you can purchase shares of those companies, and the money that's raised through those stock purchases, that's used to fund the research. And then, of course, once if a company is successful and they get a drug on the market, often they will invest a portion of their profits into additional research and development.
Um, Thank you. And I think another question is how exclusion and inclusion criteria determine are determined in different diseases like Parkinson's.
Yeah, yeah, yeah. That's a good question. Um, so when a study is being designed, it's written up into what's called a protocol, and the protocol describes your study design, like we described. Is it a randomized, double-blind, placebo-controlled study? Um, how many treatments are going to be involved? How long is the study going to be? Is it going to be in healthy volunteers, or is it going to be in patients with the disease of interest? Um, are you looking at male and female adults, or are you restricting it to a specific subpopulation? Are we going to look at only elderly patients? Um, so part of the criteria that you're developing for your study is who are we going to include? Okay, let's say we're going to include male and female patients age 55 and above. We're going to say if they have normal cardiac function, if their laboratory tests are normal, um, those are important characteristics. And we're going to exclude patients who have been receiving a dopamine product in the last 6 months. We're going to exclude patients who have any particular side effects that you don't want to be assessing in your study. So you're kind of going through all the different characteristics you do and you don't want to see in your patients. And as I mentioned, you're typically defining a pretty narrow set of characteristics. It's actually hard for patients to meet all of the criteria to participate in the study. And the reason for that, again, is you want to remove as many variables as you can that will make it hard for you to interpret your data. So hopefully I'm getting at the question in terms of what the person was asking in terms of defining criteria, but typically there are 10 to 20 inclusion criteria and 10 to 20 exclusion criteria that have to be met by a participant before they can be in a study.
Okay. I think that does answer the question. And then I think someone asked oh, I guess there's a follow-up question on that.
Yeah, exclusion,
If
are they
the
hypothetical
yeah.
or based? Okay. No, so they are, you will typically see that these criteria have been used in many research protocols. So often you'll see the same criteria applied from one study to the next study to the next study. So, and they're criteria that you're able to assess in the patient. So when, let's say we're doing a study in Parkinson's patients, and we want to determine who's going to be eligible. So there's a screening process, and this is all before they're actually treated with the drug, is you put them through the screening process where you assess, okay, we're going to, as part of the screening, we want to make sure that they meet these 20 inclusion criteria and that they don't meet any of these 20 exclusion criteria. So in order to do that, we need to do the following things. We need to take a medical history. We need to do a blood draw to assess their hematology and chemistry parameters. We need to uh, test their motor function. We need to assess their quality of life. There may be all sorts of assessments you're doing over typically a 2 to 4 week period to assess whether they're eligible for the study. And then if they meet all those criteria, then they're allowed into the study and they can receive the drug.
Okay, and then I think we have our last two questions.
Sure.
Um, so for about the financial decision, like if like an investor chooses to invest or not, how do they decide that? Like, is it through like some kind of they need some kind of metric or something before they decide to invest?
Yeah. So typically, for example, a venture capital firm, they will do what's referred to as due diligence on a company. They will, and they're often very well read in terms of a particular area of research and a disease area, and they know about the market, they know the competitors, they know typically what patients are receiving. And so it's a very well-informed decision that they're making. There are industry analysts that are, these are the folks who are familiar with the different companies, the different drugs that are being developed. They're familiar with the existing market. They are familiar with the cost to develop drugs, how much typically the drugs are going to cost when they're on the market. So these analysts are the ones who are helping to inform investors as to whether they should essentially, bet on the horse, right? To put some money down and say, yes, I think this drug looks promising based on some early results that have been published. And based on what the market analysts are saying, I feel like I would like to buy some shares of this company, or as a venture capitalist, I would like to fund this research effort and then develop that relationship with researchers. Again, whether it's with a biopharma company or with someone in academia who's decided they want to help to develop a drug.
Okay, thank you. And then the last question is MPTP still used in practice to induce Parkinson's in lab trials, or if there's some kind of new approach that's being used?
I think it still is used. Sadly, I mean, Parkinson's research has not made a substantial amount of progress. I would say over the course of my career, as a graduate student, I was involved peripherally in Parkinson's disease research. That was a long time ago. And the methodologies and the treatments have really not improved substantially. You notice that the example of the drug I gave you was a drug approved in 1975, and it's still in use. I mean, that's, 50 years ago. So the fact of the matter is that neurologic conditions in particular are very hard to treat, and even more so to come up with effective prophylaxis, prevention strategies. So there's a lot of, you can look at that in two ways. One is that's unfortunate that so many studies are not successful in neurologic conditions. You can also look at that as, there's a lot of need there, and that's motivating to me. I want to work in Alzheimer's, for example, because the progress has been very slow and incremental, and we really need some breakthroughs in Alzheimer's. And I'm motivated by that challenge. So there's different ways to look at it, but I will say that having worked in industry for a long time, and not just in neuro, but most of my career has been in oncology and in infectious diseases. Neurologic conditions by far are the most difficult to have successful studies. And there's a term that's used, it's the boneyard of neurologic diseases are considered the boneyard of drug development, meaning that many, many, many drugs have failed that have been evaluated to treat neurologic conditions. So there's, again, a lot of unmet need, a lot of progress that's still needed in neuro. So hopefully you use that as motivation because the progress has been not as quick as in other disease areas.
Okay. Thank you so much. That was so interesting, everything you said. And I was wondering if like everyone wanted to like turn on their cameras for a second and then we could take a screenshot, I guess, like of all of us.
That'd be great.
I'll turn mine on. And also if anyone has any more questions, I guess they could email. Could they email you about it
Yes,
or?
yeah. So I'll send my slide deck to the association, and they can distribute
Mm-hmm.
it. And at the end
Okay.
of the deck, you saw that I have a couple email addresses. You can email me at either one of those, and I'm happy to um, answer any further questions. I know that at this point, you're exploring perhaps different areas you might want to go into in neuroscience, and I'm happy to give some guidance in terms of directions you might go in academic programs, and even beyond that in terms of thinking about, okay, well, actually, I would like to work in a not-for-profit. Can you give me some idea of how I would get into a not-for-profit research setting in the neurosciences? I'm happy to talk you through academic program options and also career path options.
Okay. Thank you. I'm gonna take the screenshot now, so I guess like
Okay.
look like it's not 10 p.m. or I guess 5 a.m. at your house.
Yeah.
Okay. Okay, I took it.
All right. Thanks, Mehek.
Thank you.
All right.
Um,
Thanks, everyone. Have a good
yeah.
rest of your day, and we'll send around the slide deck. Feel free to reach out to me.
Bye. Thank you,
Okay. Bye,
everybody.
everyone.
Thank you.
Bye.
Yeah, thank you.